Hello, everyone. Thank you for attending today's webinar, How do dbDNA Resolves the Challenge of Plasmid DNA in Genome Editing, presented by Touchlight. I'm Rebecca Kushmeider. I'll be moderating this webinar. Our speakers today are Dr. Sarah Moore, Head of Gene Therapy Discovery at Touchlight, and Dr. Elena Stoyanova, Platform Engineer Innovations Team at Touchlight. You can read their full bios on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. This webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we'll follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. Okay, now let's begin. Dr. Moore, please go ahead.
Hi, thank you. And thank you everyone for joining today. It's a pleasure to be here to present this webinar titled How dbDNA Resolves the Challenges of Plasmid DNA in Genome Editing. So first I'd like to start with just a brief introduction to the company itself. Touchlight was founded in 2007. We currently have about 125 employees, and we recently completed a $125 million fundraise, which is being deployed primarily in three main areas of focus within the company. So at the heart of it, we really are an innovation company focused on enzymatic DNA. We're always working to improve kind of our existing platform for manufacturing dbDNA, which is our trademark molecule, but also always working to innovate around the development of novel enzymatic DNA molecules with different market applications. And this really all feeds into our CDMO. So we have a company called Touchlight DNA Services, or TDS, which is currently supplying APIs and critical starting materials to the genetic medicine industry. We've engaged over 70 customers to date, and we're currently expanding our facility to enable really the largest DNA manufacturing capabilities worldwide. And then we also have a number of ongoing research programs that are really aimed to validate and advance dbDNA in the existing DNA markets, such as DNA vaccines or viral gene therapies, non-viral gene therapies. And through these programs, we really aim to develop optimized vector platforms that can be used either in R&D collaborations or as assets through licensing to third parties. And so with that, I'll just go quickly through the agenda today. So we'll first discuss some of the key challenges in the DNA industry. We'll then introduce dbDNA as an enzymatic alternative to plasmid DNA and kind of discuss how this addresses some of those key challenges. We'll present some data to demonstrate the utility of dbDNA for viral vectors and gene editing. And then we'll also introduce one of these novel enzymatic molecules I mentioned, which we call MBDNA, and which is being developed really as a donor template for gene editing. And this will obviously be followed with some Q&A. The DNA is really fundamental to the genetic medicine revolution, where it's deployed in a number of different modalities. Recently, there's been a huge growth in viral vectors, where plasmid DNA is still really the major starting material to manufacture vectors like AAV and lentivirus. Lentivirus is then often used to make CAR T cells. And in fact, there's also been a lot of growth in using non-viral methods to generate those CAR T cells, particularly through gene editing. And then, of course, in the last few years, there's been an explosion in the mRNA market, driven by the obvious COVID pandemic. And here again, plasmid DNA, or DNA in general, is still a starting material for the IVT in vitro transcription reactions to generate your mRNA. And then we've also seen a lot of growth in the use of DNA as the API itself. So this is apparent in prophylactic vaccines, particularly with the recent emergency approval of a COVID-19 vaccine in India, but also for therapeutic cancer vaccines, and indeed being delivered as the API itself for non-viral gene therapies through LNP or exosome delivery, for example. And this growth is really well represented here by the ASGCT in 2021, showing the gene therapy pipeline and this huge expansion over the last 5 years or so. And in 2021, there were more than 1,200 gene therapy products in development. Historically, a large part of that was made up with viral vectors and still is really. But again, there's this recent proliferation in non-viral modalities for mRNA, for example, for vaccines and genome editing, as well as DNA, as I mentioned, for non-viral gene therapy, as well as vaccines. And if we look a little bit closer at how this pipeline breaks down, you can see that almost 70% of those 1,200 gene therapies are made up of genetically modified cell therapies. And indeed, more than 60% of those are CAR T-cell and TCR T-cell therapies, which really, for me, drives home the significance of this market really within the gene therapy pipeline. And this is a trend that's likely only to grow in coming years with the successes of multiple approved CAR T-cell therapies now targeting B-cell lymphomas and multiple myeloma. And so the way that CAR T-cells and TCR T-cells are made, which I'm sure will not be a surprise to anyone here really, generally starts with the isolation of autologous T-cells from a patient through leukapheresis. Those cells are then activated and then transduced with usually a lentiviral vector that expresses the desired CAR or TCR construct. The cells can then be expanded, purified, and returned to the patient. But increasingly, this process is being either complemented with or entirely mediated through gene editing. So in the context of complementing therapy, you can, at the stage of cell isolation, use gene editing to knock out genes like PD-1 or the endogenous TRAC locus to really enhance the function of the cells that will subsequently be made using lentivirus. But again, there's more and more of a drive to really use non-viral methods entirely to do the introduction of the CAR and TCR, which can be done directly into the TRAC locus, thereby kind of killing two birds with one stone. And so with that, I'd just like to focus a little bit more on the gene editing aspect of these therapies, and in particular, the multiple modalities that can be used to introduce the editing machinery. So at the top here, you can see that AAV is still one of the major vectors that's being used to deliver both the editing machinery as well as the HDR templates. And this is really due to the fact that AAV does lead to quite high efficiencies of transduction and therefore editing efficiencies. But there are some limitations, of course, one of which is, for example, the limited packaging capacity that you find with AAV. So in the context of editing machinery, this often necessitates the modification of the machinery for it to be able to fit within the packaging capacity, which is about 4.5 kb. This also limits the size of the gene that you can deliver for knock-in, although this is perhaps less of a concern than for the editing machinery itself. And then whether you're using AAV as a viral vector or lenti as a vector, you're faced with the kind of common drawbacks of viral vectors, which is the difficulties in manufacturing, the stability, and also immunogenicity, which is a serious concern. And so with that, there's been, as I think everyone knows, a drive recently to move to non-viral methods. And in the case of delivering the editing machinery, this is often done through the use of your RNP complexes, so Cas9 complex with guide RNA, or indeed you can deliver mRNA directly to express these components within your cell. And for delivery of the HDR template, this is more and more being done with DNA, whether it's a plasmid DNA as shown here, or other kind of forms of DNA like single-stranded or double-stranded oligos. But no matter what modality really you choose to deliver these components, most of them or all of them really require DNA as a starting template, perhaps with the exception of the RNP complexes, depending on how you want to look at it. But really, most of these require DNA as the starting materials to make these components. And so the increase in cell therapy applications, and particularly the increase in non-viral methods for doing that, has led directly to an increased demand for DNA reagents. And so this brings me to some of those key challenges that we see in the DNA manufacturing industry today. So if we look at kind of the predominant method for manufacturing plasmid, from a high-level perspective, this starts with E. coli-based fermentation in fixed bioreactors, followed by alkaline lysis and purification by chromatography, leaving you ultimately with your plasmid, which contains, of course, your sequence of interest, but also bacterial backbone, really. And so we see a number of drawbacks with this platform. So to start with, there's quite a well-characterized lack of fermenter capacity in the DNA industry, which is leading to long GMP production timelines. And this has obvious implications for the timelines of people's programs, as well as costs. There's also a problem with DNA instability. So there are a number of sequences, or complex sequences, that are quite deleterious to bacterial propagation. Things like high-order secondary structures like ITRs, GC-rich regions, homopolymeric tracts. All of these can lead to low yields, heterogeneity within your batch, and actually even complete batch failure, which again has obvious implications for costs and timelines. And then ultimately you're left with a product which is perhaps not ideal in the fact that it contains these bacterial propagation elements, so including antibacterial resistance genes, which we know are undergoing increased scrutiny these days. But beyond those, you also have unmethylated CpG and origins of replication, which can have undesired effects on innate immune signaling pathways. And so now I'll just briefly introduce dbDNA as an alternative to plasmid DNA. So these vectors are linear, double-stranded, and covalently closed DNA vectors. With the exception of the two 28 base pair flanking sites here, which are really remnants from the manufacturing process, these vectors are entirely comprised of your DNA sequence of interest, so no extraneous sequences whatsoever. These are enzymatically amplified in a fully cell-free process, which affords a few advantages in that we are able to introduce highly complex sequences into these vectors and manufacture them with high reproducibility outside of the selective pressures of E. coli. And we've also have experience making a wide range of sizes of dbDNA. We currently make anywhere from 1 to 20 kb quite routinely. So the way that dbDNA is made is it starts off first with a traditional plasmid with your sequence of interest flanked by these protelomerase binding sites that are shown here in yellow. This is denatured, and then 529 polymerase is added to the reaction, leading to rolling circle amplification. This leads to strand displacement and then the formation of long single-stranded DNA concatenamers that basically alternate between your gene of interest and the plasmid backbone. Then second strand synthesis reactions generate your long double-stranded DNA concatenamers, as you can see here. Then protelomerase is added to the reaction, where it is recruited to the protelomerase binding sites, leading to cleavage and end-joining reactions that leave you ultimately with your covalently closed doggybone DNA, as well as some backbone impurities. And so first process of column chromatography removes your enzyme impurities, and restriction enzymes and exonucleases are added to digest away the non-target material, so basically your plasmid backbone material, leaving you with just your doggybone, and then further purification removes the enzymes and exonucleases, leaving you with your final pure dbDNA population. This is then filtered through a 0.2 micron filter and dispensed under laminar airflow. In terms of our GMP manufacturing capabilities, we currently have 4 established suites. These sit in a very small footprint of about 9 square meters, and they contain only very simple benchtop equipment. They use only single-use contact materials. And each suite can produce upwards of 100 grams per year in what we call our mobile scale process, which is derived from upstream reactions of up to 5 grams in a complete manufacturing process of 5 days. We're always working to improve that output, and indeed we are developing an industrial scale process which is aimed more at kind of commercial production and late-stage clinical production, which has an output of about 5 to 7 times that of our mobile scale process. And as I mentioned at the beginning, we're also increasing the size of our facility quite considerably. We're adding another 11 suites, which are being designed to be quite flexible to accommodate production scales of anywhere from a few mgs to hundreds of grams, which should enable us to have a multi-kilogram capacity by the end of the year. And so taking these factors in mind, we really think that dbDNA is well placed to address some of the challenges that we've just discussed in the DNA manufacturing industry. So to start with, our manufacturing platform deploys very simple equipment in a small footprint. This enables gram scale batches in days, which can reduce your production timelines by up to 80%. 529 is the enzyme that we use for manufacturing, and this is an enzyme that mediates rolling circuit amplification and is therefore very well placed to amplify through very complex sequences like ITRs, polyAs, GC-rich regions. And ultimately, we're able to produce with high consistency these unstable sequences that previously have been quite difficult to make in plasmid. And indeed, the quality of the doggybone that comes out of our process really is a reflection of the quality of the template that goes in. And then I think it goes without saying that the removal of bacterial sequences is a benefit both from the regulatory perspective, but also from the point of view of improving the safety profile of your product. So I talked a little bit now about how doggybone addresses those general challenges, but I also wanted to point out a few ways in which it addresses some of the specific challenges that are faced by the gene editing industry. Particularly when we're thinking of non-viral methods, one of those challenges is the low transfection efficiencies that you can be faced with when using plasmid DNA. And one of the factors that affects that low transfection efficiency is the large size of plasmid DNA. And so dbDNA, being by nature much smaller than plasmid, has an intrinsic copy number advantage. And that means that just by default, you can deliver more copies of your molecules, so more copies of your HDR template, for example, using the same amount of DNA as you would for plasmid. But beyond that, we actually find that dbDNA can have an improved transfection profile compared to plasmid. So the data that's shown on the right just basically shows cells that were transfected with 4 different constructs, either as plasmid or dbDNA, under mass match conditions, where you would expect to have perhaps a 1.5-fold advantage for doggybone just based on the size difference. But actually what we see is a 3 to 4-fold copy advantage for dbDNA constructs, suggesting that it's somehow getting in the cells more efficiently, whether that's due to the smaller size, the linear nature. We don't have the answer to that yet, but we do see this. And so this probably potentially provides the possibility of enhanced editing efficiencies at lower doses. Then another factor affecting that low transfection efficiency is the immunogenicity of DNA, particularly double-stranded DNA. And this is especially apparent when you're trying to transfect delicate primary immune cells, for example. And we find that doggybone has a reduced immunogenic profile compared to plasmid. So first of all, there's no activation of TLR9 whatsoever. And then we also have found that it has a reduced inflammatory profile in certain cells, such as iPS cells, for example. So this plot on the right, or this heat map on the right, basically shows the activation of interferon signaling markers in iPS cells that were derived using either plasmid or doggybone. And this is compared to the negative control here, which is embryonic stem cells. And you can see that doggybone in the middle, those cells have a much more similar profile to those of embryonic stem cells than those generated using plasmid. And this is interesting when you think about the fact that these cells have been passaged many times and therefore no longer contain this, well, presumably no longer contain this transiently transfected DNA. And really driving home that point that those early events can have a significant impact on your final cell product. And so together, this reduced requirement for input material and reduced immunogenicity provides the potential for reduced toxicity, which really should translate into improved editing efficiencies. And I mentioned also earlier that we have a number of programs to validate dbDNA in kind of all the addressable markets. So I don't have time to talk about all those today, obviously, but I'll focus just on those that are highly relevant to gene editing. So here's a few data sets. On the left here is shown a representative production for recombinant AAV. I believe this was AAV9, where we're using doggybone or plasmid to manufacture. And actually here we're using only 60% of the input DNA using doggybone as you do with plasmid. And you can see that you get similar to higher yields, in fact, both at the level of your genome-containing particles measured by PCR, qPCR, as well as your total particles here. Equally, with lentiviral vectors, we're able to make equivalent infectious titers. And so this is done here in the context of CD19 CAR-T lentiviral vector. And again, we're using only 70% of the input DNA here and achieving similar infectious titers. And then finally on the right is a plot showing the use of dbDNA as a starting material for making mRNA. And here we're using mass matched conditions. And you can see that we actually are able to produce higher yields of mRNA with comparability at the level of potency, integrity, etc. And then here's some data demonstrating the use of dbDNA as a donor template for gene editing. And these are primary T cells that were transfected with an RNP and then either a plasmid or doggybone GFP donor at a kind of a range of concentrations from 0.25 picomole up to 2 picomole. And if you look at the higher concentrations here, you can see that, from day 2, 4, and 8, that plasmid does still outperform doggybone somewhat, with a maximum of about 35% knock-in efficiency for plasmid compared to about 28% for doggybone. I think it's worth noting here that the conditions used for transfection had really been optimized for plasmid DNA. And we do find that doggybone and plasmid can require different conditions for transfection. And so we feel quite confident that with some optimization that this can be, basically we can increase the editing efficiencies of doggybone at the higher concentrations. But what is interesting, and something which we've observed a number of times now, is that as you down titrate doggybone DNA, the editing efficiencies are much more stable than for plasmid. And so you can see here at the lowest concentration with plasmid, you're reaching only about 5% knock-in, where with doggybone, we're still holding at about 15%. And again, this is something that we've seen multiple times in different contexts. And we've also seen this comparison, the similar observation made relative to not only plasmid, but also minicircle DNA. So really suggesting, again, that there's potential to have higher efficiencies of editing at lower concentrations. And again, we're still working very hard to optimize the conditions for transfection to boost this editing overall. And so with that, I'm actually going to hand over to my colleague, Elena, who will be introducing MBDNA, which is, again, one of these novel molecules that we're working on to use as a donor template for editing. Take it away, Elena.
Thanks very much, Sarah. So I'll present to you one of Touchlight's novel platforms, which is specifically designed for gene editing, and it's a novel molecule called MBDNA, or Megaboop DNA, which is essentially a single-stranded closed CRISPR HDR donor that we at Touchlight have an IP on. So it consists of a long single-stranded loop that encodes for a template for CRISPR gene editing and a short double-stranded hairpin forming this light bulb-like structure, hence the name Megaboop. So within the base of the stem, Megaboop contains a nuclease target site, which in this case and for the purpose of this talk is going to be Cas9, but it can be any targeted nuclease really. So Megaboop gets cleaved by Cas9 and releases the single-stranded HDR donor for CRISPR. So why is Megaboop such a good gene editing template and significantly better than the existing technologies? Well, first of all, it is single-stranded DNA, making it more efficient, less toxic, and more specific than double-stranded DNA. It is a non-viral gene editing technology that has the capacity to encode long DNA sequences, which overcomes the issue with long single-stranded oligosynthesis, which is both complex and can be very expensive. This also allows us to beat the packaging capacity of viral vectors such as AAV, for example. The closed structure of Megaboop provides increased stability in the cell over single-stranded open oligos. Megaboop also can offer enhanced specificity because in theory it is extremely on target because the single-stranded donor will only be released in the presence of Cas9. And because the target site on the Megaboop is the same as the target site in the genome, only cells that already have the Cas9 RNP that's aimed for knockout will also have the single-stranded donor released, which can significantly reduce off-target effects. So another advantage of the Cas9 target site on the Megaboop could be its facilitated trafficking into the nucleus and additional protection from degradation in the cell, which is something that's been shown to improve AGR for both double-stranded as well as single-stranded CRISPR AGR donors in a couple of studies from the Marston's lab that are cited here. So in our first proof of concept study, we're not only able to show that Megaboop can successfully introduce gene length knock-ins in HEC293 cells, but that it can do so more efficiently than other HDR donors. So for this study, we used a donor that encodes for a red fluorescent protein, and you can see here a diagram, the Megaboop encoding like a red fluorescent sequence, and just a reminder, it contains this Cas9 target site at the base of its stem. So you can see that some of these HEC cells are successfully expressing the red fluorescent protein following editing with Megaboop. So before I show you some flow cytometry data comparing Megaboop to other HDR donors, I would just like to point out that this is very early on data where we hadn't really optimized the Cas9 targeting or the editing efficiency in these cells with this donor, and the DNA donor amounts we used were very low molar amounts, which is why the AGR rates you're going to see are not very high. But what is important is that Megaboop performed better to the other CRISPR AGR donors when compared in these suboptimal experimental conditions. So first of all, we compared Megaboop to an SSODN or single-stranded oligo at 3 different concentrations. Megaboop performed better than the SSODN at every concentration tested. Megaboop also maintained more stable editing efficiency over a concentration range compared to an SSODN, presumably because of its improved stability in the cell. So whereas a SSODN effect drops down significantly when you reduce donor concentration because it gets potentially rapidly degraded at low concentration in the cell, Megaboop maintained more stable knock-in rates even at the lowest concentration tested. So then we wanted to test that the Cas9 target site in Megaboop is important for its functionality, which is why we compared a Megaboop to the exact same Megaboop that lacks a Cas9 target site. So the Megaboop that lacked the Cas9 target site performed consistently 4-fold worse than the original Megaboop, demonstrating that the Cas9 target site is important for Megaboop's functionality, and it's important for efficiently mediating HDR with Megaboop. So when compared to double-stranded donors, we first compared Megaboop to a standard plasmid DNA, and at the concentrations tested, plasmid DNA didn't really work at all compared to Megaboop. So then we compared Megaboop to a plasmid with Cas9 target sites that were flanking the donor DNA sequence on the plasmid that could help with nuclear trafficking as well as releasing the double-stranded CRISPR donor in the cell that could aid editing efficiency. So this indeed helped improve editing efficiency with plasmid, but still didn't manage to beat Megaboop. So because Megaboop works so well in HEC cells, we decided to move on and test it in primary T cells, which is why since then we have been working on the construction and testing on a couple of track-targeting Megaboop constructs for T-cell editing. So these are based on a published and well-verified CD19 CAR sequences. These designs are improved compared to the Megaboop that we used in the HEC cell study. We have 800 base pairs homology arms and a very well-verified guide RNA sequence. We have 2 constructs, one encoding for EGFP for ease of screening, and one that encodes the actual CD19 CAR. We have several ongoing collaborations, and in combination with some in-house testing, we have initial data of these Megaboop, one of these Megaboop constructs in primary T cells and some in iPSC cells coming soon. So the first evidence we got that Megaboop works and works well in primary T cells comes from our partner Syntago, where in this pilot study, they compared a range of 0.25 to 2 picomole of the track EGFP Megaboop donor and an equivalent plasmid DNA donor. So they managed to successfully introduce increasing levels of knock-in using increasing levels of donor concentrations. So looking at the editing efficiency only, at first it seems like Megaboop is outperformed by the plasmid DNA donor here, but that is not until we looked at the viability data. So this is a plot showing the live cell percentages as they're measured not by live dead stain, but via gating in the forward scatter versus side scatter plot, which is why the percentages here are not very high, but this is true across the board for all the samples, including for the mock control, which are just cells electroporated with a pose without any cargo DNA. So what we can immediately see is that not only is plasmid DNA a lot more toxic than Megaboop DNA, but increasing concentrations of plasmid DNA decrease cell viability significantly, as opposed to Megaboop DNA that maintains our cells happy and alive at all concentrations tested, even at the highest 2 picomole, producing live cell percentages as high as or even higher than mock. So this is extremely important because if we actually take this exact same data but look at it in a different way, and we look at the amount of cells, the cells that have been at the same time alive and edited as a proportion of the original population, Megaboop would produce a much higher live edited cell count at the end at 2 picomole compared to plasmid DNA. So now this is extremely important, of course, in ex vivo cell therapy context, where you can only harvest a limited number of cells from a patient. So, for instance, in this example, if we had collected 100 million T cells from a patient, 2 picomole of plasmid would produce about 10 million live edited cells at the end, as opposed to Megaboop, this would have produced 14 million live edited cells, nearly 50% more. So because of the improved viability, plasmid DNA is significantly outperformed than Megaboop DNA. And because we still haven't seen a drop in viability, we can only envisage that with increasing concentration of donor DNA, these numbers would only go up for Megaboop and down for plasmid DNA. So then the next thing we did was to test an even higher concentration of Megaboop DNA. So we looked at between 2 and 6 picomole Megaboop. So with this, we managed to successfully increase knock-in by increasing donor concentrations while keeping our cells extremely, incredibly alive. So you can look at the viability data on the right-hand side, and you see that the cells edited with even 6 picomole are about 95 and above percent alive. So this is absolutely key because it suggests that we can use increasing concentrations of donor DNA to increase knock-in while not sacrificing our cells' viability. So this is something that we are currently working on and exploring. We would like to test the range to keep increasing the donor concentration with Megaboop to increase the knock-in, and we'd like to test the concentration range in which we can still keep doing that while keeping our cells extremely viable. So with this, I'm going to go back to our Megaboop products that are available for third-party testing. And I showed you data for our EGFP construct, and we'll soon have data for the CD19 CAR. We are also able to make, and have done so, custom Megaboop sequences where we can encode a donor sequence of your choice, and as well as a different target nuclease to Cas9. So with this, I'm going to go to our summary slide, and Sarah and I essentially presented to you two of Touchlight's platform, our dbDNA, our core platform, where we showed you we could rapidly enzymatically produce linear DNA at scale, GMP grade, and dbDNA performs comparably to other DNA format in genome editing, and it can work efficiently as an HDR donor template. I also talked to you about Megaboop DNA, which is our novel molecule that is specifically designed for gene editing, and it shows significantly improved viability compared to plasmid DNA, showing the potential to increase donor concentration, to increase knock-in without sacrificing our cells' viability. So thank you very much, and we're happy to take questions.
Now let's move on to our Q&A, and I'd like to introduce you to Tommy Duncan, the Chief Business Officer, who will be joining us for this portion of the webinar. There is still time to submit questions using the Q&A tab to the left of your screen. We have a lot of great questions already. We'll try to get to as many as possible. For the first question, how does the fidelity of Phi29 compare to plasmid?
Hi, yeah, I can take that. So the published error rate for Phi29 is 10 to the minus 7, which is obviously extremely low. But we have a team that's dedicated to kind of using NGS to validate changes in our process, for example, to make sure that the fidelity of amplification isn't affected by any modifications to the process. And through this and through some other kind of research programs, we've found that when we use NGS, the error rate that we see for both plasmid and dbDNA throughout kind of what we call normal sequences is around 10 to the minus 4. And the error rate for NGS is around 10 to the minus 4 to 10 to the minus 5. So that's really kind of in the weeds of what you can even pick up with any sort of accuracy, really, by NGS. So in terms of kind of normal sequences, we would say that there really isn't any detectable difference in the fidelity of amplification. That maybe doesn't do justice to the platform, though, because where we do see differences is, again, in these complex sequences, right, where in particular ITRs, where with plasmid amplification, you can often get deletions, insertions, truncations even. And here we see that, we clearly see the benefits of our platform in the fidelity of the amplification of ITRs. You know, we've produced and sequenced over 200 constructs, obviously not all ITR containing constructs, but generally speaking, all of our constructs are fully sequence verified before being released. And to date, we have yet to find a mutation that was derived from the actual manufacturing process itself. So, we would say that the fidelity is very, very good using Phi29 in our platform.
The next question. What do you expect the bridging requirements to be for switching from plasmid-derived product to the dbDNA-derived product?
Yeah, we have quite a bit of regulatory interaction that we've been through over recent months and years on this particular topic, because it's an important topic for Touchlight. And we operate across industries, as Sarah had introduced earlier, with producing dbDNA materials as a critical starting material, as well as an active pharmaceutical ingredient. And the feedback that we've had from regulators, inclusive of European and American regulators, is that when switching between plasmid-derived products, for example, viral vectors or mRNA, and dbDNA as a critical starting material, then the expectation is that they would want to see analytical comparability between the materials that are made with both of those different starting materials. So obviously that's related to then getting an equivalent specification and an equivalent product that is produced. But what it means also is that there's no need to do any kind of bridging studies related to animal studies or tox or anything like that. So very positive on that front to date.
The next question, to what scale can you manufacture?
Yes, Sarah introduced earlier the manufacturing suites that we have. They're very small suites with a benchtop footprint, but we are able within that footprint to make quite a significant amount of DNA. The routine manufacturing size or manufacturing scale at the moment is up to about 5 grams. We have an ongoing program for scale up as well, which will probably have a batch size of around about 40 grams. But at present, anything in the GMP scales, anything between 100 milligrams and up to 5 grams is around about the scale that we're producing.
Next question. Can you make long sequences such as helper constructs?
Yeah, so I can take that. As I mentioned kind of earlier, we quite routinely make, I mean, theoretically, there's actually no limit really to the size that we can make. But of course, the efficiency does go down if you get to really, really, really large sizes. But we quite routinely make up to 20 kb constructs, which does cover most of the helper constructs that you can imagine. I mean, some of the very large AAV helper constructs are around 18 to 19 kb. So we sit well within that, and indeed we've made helper constructs very efficiently for multiple AAV programs. So that certainly isn't a concern.
Next question is, can you make complex sequences such as ITRs?
Yeah, again, so 529 is quite an impressive enzyme, really. It has mediates rolling circuit amplification, and so therefore it's one of these enzymes that has really strong kind of debranching activity. It has very strong single-stranded DNA binding activity, which means it's very able to unwind DNA. It's highly processive, and it has intrinsic proofreading activity. And so it is very well placed to amplify through complex secondary structures like ITRs, again, with quite high fidelity. And, we do have quite extensive experience making ITR structures, as mentioned previously. And these are all fully sequenced as part of the standard QC release process. And again, we've not found any mutations in doing that. So I would say that, yes, we're quite capable of making ITR constructs.
Next question is, how does dbDNA perform in an in vivo context?
Yeah, well, we've tested it in numerous in vivo contexts. So in some of the programs that I wasn't able to talk about today, and we've delivered it in very different modalities. And generally speaking, we see that it performs very similarly to plasmid, or even better, depending on the context. In some contexts, we see clear dose-sparing effect using dbDNA, as I've alluded to in the earlier presentation. But whether we've delivered using LNPs systemically or liposomes directly to the muscle, nanoparticles systemically as well for different programs, or obviously naked DNA delivered by IMEP for the vaccine programs, both prophylactic and therapeutic. In different animal models. And again, we always see that dbDNA performs similarly or better, really, compared to the other modalities that we test.
Our next question is, what type of chromatography are you using as a first and second step?
Yeah, I can take this one. The chromatography method that we use for both steps is the same method. We use a commercially available resin, and we perform the purification in flow-through mode. So obviously you're binding the impurities such as the proteins, leaving the dbDNA there. So the purity profile of the dbDNA is actually exceptionally high, because there are really proteins of known identity and nucleotides that you need to clear from the reaction. And it's done most efficiently in flow-through mode, in our experience.
The next question, is there a limit of size for dbDNA construct, and how is the yield/purity affected by long sequences coding for mRNA poly A tails compared to recombinantly expressed constructs? So
I think we briefly covered the size factor in the previous question regarding ITR. So again, we can make up to 20 kb very routinely. As far as poly A tails, so routinely we make about 120 base pair poly A tables. We've done up to 150 on behalf of a third party, and actually in-house we've tested up to 200 base pairs. We find that it performs very nicely compared to the other formats, because again, the process really is quite agnostic to sequence. So we don't see a huge effect on efficiency of production with the introduction of these long poly A tails.
Next question. What are the main product-related impurities using the dbDNA technology, and are truncations a frequent issue?
Yeah, the product-related impurities, as you can imagine, would be those proteins of known identity that are the enzymes used in the manufacturing process. So it would be the polymerase, the protelomerase enzyme. And in terms of our specification, we do measure protein content, and we have a specific ELISA on the protelomerase enzyme, which is used towards the end of the process and in the highest quantity. The question around truncations, well, that's covered by the downstream purification process of dbDNA. The dbDNA itself is impervious to exonucleases, and there is an exonuclease polishing step within the downstream of the dbDNA production. So what that means is that we eliminate anything that isn't a double-stranded covalently closed molecule. So anything that may be an impurity as a single-stranded or a truncated product is eliminated from the reaction. So again, it's a very stringent way of clearing those particular impurities.
Okay.
Next question. How are these MDDNAs generated?
Yeah, I think that's MBDNA. Yes, so we're not disclosing the production method of Megaboop DNA just yet, but we obviously, as you can expect, are an enzymatic nucleic acid synthesis company, and so we do apply the types of methods that you would expect of a company like us to produce our MBDNA.
Do you have plans to eliminate the use of plasmid DNA to generate dbDNA?
Yes, we do. We have a number of ongoing programs, as you can expect. We do see a future entirely in the absence of that plasmid-based critical starting material. Obviously, it benefits from being a clone at present, which is extremely high fidelity from which to amplify from. But we have made significant strides on that next generation, which is the initiation of the reaction from fully synthetic starting materials.
Next question. How long does it take to manufacture MBDNA and dbDNA versus standard pDNA?
Yeah, so the production process of dbDNA that Sarah described in the animation during the webinar really shows a process that at GMP, at multi-gram scale within those suites, is a 5-day process. So there are additional steps that you might perform as well. For example, when we're making template for IVT, we would then have a linearization or what we call a de-ending step, which would then take an additional day or so. So the actual production in the GMP suites is 5 days. So the QC release is very quick. It's about 2 weeks. And obviously at the front end of the process is the preparation of that plasmid template. But in terms of turnaround times of GMP quality dbDNA, it's significantly quicker than being able to do this with fermentation-based methods. Also, we don't require a GMP cell bank for our critical starting material, which at present is a plasmid, which also can improve the timelines to turning around the material. In terms of the Megaboop production process, it's obviously more of a research process right now, so that's not fully locked down. But using enzymatic methods, obviously, it's not wildly dissimilar to the dbDNA process.
Does the MBDNA allow for improved development of CRISPR-Cas like Cas12 and 12A?
Yes, so I'll take this question. Yes, definitely. So as I said in the presentation, the Megaboop, so the Cas9 target site in the Megaboop can be for any targeted nuclease. So it doesn't have to be, it's not restricted to Cas9. And we have worked on different designs with especially Cas12. So yeah, it is definitely compatible, and it can work with those systems.
Is GMP quality for pDNA, dbDNA, MBDNA necessary?
We would say yes, and I think the quality of the material produced can be dependent upon the end use application. Obviously, for research grade, you wouldn't expect to be using GMP quality materials. We have a lot of different use cases for the dbDNA. For example, we've made GMP material for IVT products that are themselves critical starting materials for cell therapy in a genome editing context. So that is some number of steps away from the dbDNA itself being the API. But typically, we apply exactly the same process and materials to production in that context, regardless of what the end use is. We do have a grade called Smart GMP, which we produce, which essentially has a slightly different documentation burden versus GMP dbDNA, but really the material that's produced is pretty much identical.
Our next question is, have you ever compared the knock in efficiency between single-strand oligomer and MBDNA?
So the answer is yes. I did talk about this briefly in the presentation. So we have compared Megaboop to an SSODN in hexels, and Megaboop performs significantly better. So which is probably because of its improved stability in the cell. We haven't really investigated the method, but it definitely does work better.
What quality standard is Touchlight manufacturing against to claim cGMP? Is your quality system based on ICH-Q7?
Yep, so the answer's in the question there. The quality system is based on ICH-Q7, so that's the standard that we use.
Does Ki by MBDNA require more RNP compared to pDNA?
So from what we've seen so far, no, it doesn't. So I understand maybe this question comes from the fact that Megaboop is bound by Cas9. So maybe maybe you would need more for knockout in the genome, but that's not what we've seen so far. So Megaboop would get bound by Cas9, but then it would get released in the cell. So I'm assuming Cas9 gets recycled essentially. So yeah, you can use the same amount that you'd use with plasmid DNA. That works fine.
Are there any special considerations to take into account when working with dbDNA, and how much development should be expected if transitioning to dbDNA?
So
go ahead, Sarah.
Yeah, sorry, I was just going to talk about kind of the technical aspects of working with it, in addition to the regulatory aspects we spoke about before. But I mean, basically, it is a slightly different molecule than plasmid, for example. So because of the smaller size, the linearity, the fact that it's not supercoiled, we do find that you have to work with it a little bit differently in some contexts. This usually means some simple optimization, like in vitro experiments to evaluate transfection efficiencies based on different ratios of transfection reagent to DNA, for example. The total input DNA is also something to be considered. So elements like that, if you're working with viral vectors, it's often the case that you need to maybe adjust the ratios of the constructs going into the productions to get the equivalent amounts of titer. So just small changes like that. But once these things are established, they're very translatable between different vectors, for example, or different applications. And so I would say that, the requirements, really, the development work that would be required is really mostly that, some moderate DOE studies to evaluate parameters like transfection reagents, dosing, and that's really it, to be honest. So nothing enormous. I mean, really anything that can be done with plasmid can be done with dbDNA, just slightly different parameters.
What is the fidelity of MBDNA sequences percentage-wise, and how is this measured?
So that is similar to the one for dbDNA, and I think Sarah mentioned in the talk we've measured that with NGS, and yeah, this is how you can assess it. But yeah, it is comparable to our dbDNA.
Next question, is dbDNA cheaper to manufacture than pDNA?
Yeah, I can take this one. So dbDNA obviously is produced in an enzymatic process. So I think that it's important to say that the cost structure of making dbDNA to GMP is very different from making pDNA. pDNA is predominantly the cost of capital and labour. It's quite a labour-intensive process, and it takes a relatively large footprint and some significant fixed equipment, none of which is required for dbDNA manufacture. So it obviously, we have small footprint suites, we have relatively minimal equipment and benchtop equipment really that's required. And so that does give us certain advantages, partly of going up and down in scales very precisely. In terms of scale production of dbDNA versus plasmid DNA, because we're able to produce our enzymes at very large scale, there are significant scale economies there. So we do expect that at scale, dbDNA will be pretty competitive with plasmid DNA. I think what we do see as well is when the yields of plasmid DNA are a challenge, which can be the case for very difficult sequences, then the cost tends to go up, and we don't tend to see as much variability depending on what would historically be considered a difficult sequence.
What's the size limit for MBDNA?
So there is no size limit to MBDNA. So essentially because of the way we manufacture it, we can, and it's an enzymatic process, we can essentially encode sequences encode as long sequence as we want, which is one big advantage that we have over single-stranded oligosynthesis, which is usually size limited and is very complex. And we also, another advantage is that we can overcome the packaging capacity of viral vectors using our Megaboop technology.
Next question. How are you working to optimize editing efficiencies with MBDNA?
So, I'll take this. So there are quite a few things that we are that we are exploring. So we haven't really worked on optimizing the editing efficiency at all. So everything that I've showed you so far is almost like only proof of concept. So we're just starting to explore this space. And some obvious things that we can work on is varying the homology arm length, switching, looking at different antisense or antisense strands. So there's loads that we can do, different targets, and so on. So yeah, this is something that we're really, really excited to explore.
This will be our last question. We have had a lot of great questions today, and we couldn't get to all of them, but we will try our best to get back to everyone who submitted personally after the webinar. So for our last question, do big pDNA producers feel threatened by dbDNA and all synthetic DNA?
I think, well, obviously our ambition is for dbDNA and our other DNA molecules that we are inventing and developing to become a very well-utilized enabling product across the industry of genetic medicine. And it does certainly present certain advantages versus pDNA in a number of different spaces. And it depends what modality a particular party is working in as to what that proposition is using dbDNA. Obviously, we are seeing people who have specific problems with their plasmid DNA supply, whether it's something to do with the mutagenicity of a difficult sequence, such as a poly-A tail or an ITR, tend to gravitate towards our platform because it is agnostic of sequence as far as we can see. But equally, we do have the advantage of quicker turnaround times to GMP as well with our platform, which should be a more general benefit, especially with a molecule that we're producing that has comparable results in most places and eliminates that bacterial sequence that can be a regulatory issue as well. So yes, of course, it's our ambition to penetrate this market that is typically utilizing pDNA, but I can't sort of comment too specifically on the question itself.
Thank you all for attending this Fierce Biotech webinar and submitting so many great questions. I'd like to thank our speakers for participating and Touchlight for presenting today's webinar. This webinar has been recorded. You'll be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.